Good morning, everyone. My name is Liza Moskowitz. I am a senior studying public relations from BU's College of Communication. And here I'm going to talk about my research project, which looked at why undergraduate males are not studying public relations. The question is, what is public relations? Why do I study public relations? Is it a degree where I learn how to write a press release? And long story, no, it's not a spin doctor. I am not learning how to be Samantha Jones from Sex and the City. PRSA, which is the Public Relations Society of America, defines public relations as the strategic communication strategies that really build mutually benefit relationships between organizations and its publics. So the question is, why aren't males wanting to do that? My interest really spurred from looking at my public relations classes here at BU. All of my PR professors are males, but when I look around, it was a miracle if there was one boy in my class. In 1986, a progressive study came out that deemed public relations as the next velvet ghetto, meaning that this industry was going to become like nursing or teaching, a profession that was severely underpaid because it was dominated by female professionals. 30 years later, in 2012, an Australian research firm did the exact same study and deemed public relations as a pink ghetto, as a profession that was heavily dominated by male executives, but all entry-level positions were females. There have been no national studies, though, on looking at why males aren't studying public relations and then entering the field. So through my national study, I surveyed more than 60 higher education institutions, undergraduate males who are studying PR, studying communication majors, but aren't PR, such as advertising or marketing, as well as non-PR majors, and looked at their perceptions of public relations as an academic field and a profession. It's really interesting to see their reasonings why they decided not to study PR, and then to see the knowledge discrepancies between students who are studying public relations and students who are not, really giving way into explaining those qualitative free responses. My poster is on the far left. Hope to see you there.
So my name is Lulu Shu. I'm a human phys student in Sargent, and I did my project under Professor Mohammed Zaman in his global health lab. So my project is about the dangers of fake drugs, specifically for tuberculosis. Tuberculosis is the second deadliest infectious disease in the world behind AIDS. And part of the reason is the treatment is very stringent. You need to take the two most important drugs, isoniazid and rifampicin, at the same time, for 6 to 9 months too. If you take only one, or if you take your medications responsibly, but one of the drugs is substandard, then you'll quickly develop drug resistance. And then you'll have what's called multi-drug resistant tuberculosis. And it's very expensive to treat, and it's often deadly. So countries around the world are trying to regulate substandard drugs. Before they can do that, they need the technology. And current methods are often inadequate, or they're too expensive for resource-poor countries that are struggling the most with tuberculosis. So our lab is designing a microfluidic chip that can cheaply and accurately detect substandard drugs. The design is, we'll design a probe made of DNA, and you mix it together with its specific drug, and they'll bind and fluoresce, and then we can quantify the amount of fluorescence and figure out how much drug you have. So my specific role in the project is I made a probe with an existing molecule for the TB drug isoniazid to test this theory. And then I found that the protocol we were using to make these specific DNA probes was not optimized, and we're losing a lot of possible DNA candidates in the process. So I just optimized the protocol and made it more effective and more efficient. Thank you, and I hope you check out the details for my poster.
My name is Melissa, and my advisor was Professor Jonathan Zatlin. And my project was investigating the situation of utopian literature. I did that through a few methods. I read a bunch of utopian fiction from all genres and all times too, and I found myself getting really interested in contemporary utopian fiction. And I investigated that more, and 3 research questions kind of popped up for me. Like, what is the definition of contemporary utopian fiction? Why is it so popular in young adult and teen fiction, and what purpose might it serve in wider society? So I tried to answer those questions really directly in a nonfiction essay, and I also contributed to the genre myself by writing my own story, a part one utopian fiction story. And I hope you guys will come visit my poster and learn some more about it. Thanks.
My project is a molecular characterization of the BMP2 MEF2A pathway, which is a signaling pathway in heart cells. So this is basically what tells heart cells to go into one type of cell or another type of cell. So obviously, in utero, your heart is not a functioning heart yet, and then when you're born, hopefully it works properly. So this is how your heart cells know to do what they're supposed to do. And so obviously you have the same DNA in all the cells in your body, yet your liver's a liver, and your arm's an arm, and your eye's an eye. So what these molecules do is they kind of tell what part of the DNA do we want for specific areas. And the reason that this is important is that some of the models that we see in lab are mimicking human conditions, so congenital birth, heart defects. And so by investigating this pathway, we kind of hope to further elucidate potential solutions to these problems. Also, it's implicated in coronary artery disease. So I know this is a lot of science, but it's basically how your cells know to go from one stage to another, and there are proteins within these cells. Hopefully everyone remembers what proteins are. And it's a signaling pathway in heart cells. So I have a diagram on my poster out there, and some cells you can look at if you're so inclined. So hopefully it makes sense to everyone out there.
Hi everyone, my name is Sam, and over the past 4 years, I've had the opportunity to study economics and international relations, and I'm excited to continue my studies next year as I pursue a Master's of Public Health. This interdisciplinary academic approach allowed me to take a sort of holistic perspective in looking at why the 2014 West African Ebola epidemic was able to reach the epidemic proportions that it did. As of epidemiological week ending April 22nd, 26,101 total cases have been reported, leading to 10,845 deaths in West Africa. To put this in perspective, since Ebola was first discovered in 1976, a total of 2,500 total cases have been reported and 1,400 deaths. So clearly this epidemic was on a much greater scale than all previous epidemics. So I took a systemic approach and looking at the political, economic, and societal factors within Guinea, Liberia, and Sierra Leone, the West African countries most affected by the 2014 outbreak, and determined that it was systemic features within these countries in conjunction with the slow and inadequate international response, which allowed this epidemic to reach such great proportions. I also determined that although Ebola is a very frightening disease to a lot of people, a fact that's exacerbated by the media, Ebola is not the disease of most primary concern in either West Africa or in sub-Saharan Africa in general. And for this reason, we really need to strengthen the healthcare systems within not only West African countries, but throughout sub-Saharan Africa in general to curb not only future Ebola outbreaks, but all infectious disease outbreaks in general. Thank you.
Hi everyone, my name is Lindsey Smith. For my project for the Keystone Symposium, I conducted a year-long project under the advising of Les Kaufman in his marine biology lab. So over my time spent here at BU, I've developed a really strong interest in environmental policy, and that's why I was interested in this project. So just to give a brief introduction, so my project was specifically focused in the Gulf of Maine on Stellwagen Bank. For anybody who's not aware, New England and Massachusetts especially really depends on the functionality of this ecosystem, specifically for our economy. We have the whale watching industry that brings in tens of millions of dollars a year. We have so many different fishery industries that have been showing substantial crashes over the past 50 years or so. And this is really calling for more specific ecosystem-based management. It goes back to that whole issue of environmental policy of, okay, well, do we want to take all that we can now and not worry about the future, or do we want to plan sustainably so that this system can continue to support this economy in the future? So what my specific focus of this project was studying the diets of great shearwaters, which is an abundant bird in the ecosystem, specifically looking at its prey guild. So the unique thing about Stellwagenbank for this northern latitude is its productivity and the number of species that it supports. And so the interesting thing is that there's a prey guild in the ecosystem that is made up of a whole bunch of different species of fish that kind of take up the same trophic level, which is sort of counterintuitive in the whole competition aspect of ecology. So I was just studying how varied this seabird diet is and how specifically important each species is in order to better inform the National Ocean Policy of 2010 executive order presented by Barack Obama. So all this data is serving to support that policy so that it can be implemented effectively. So thank you. My poster's in the corner, so come check it out. All right, I'm back. I am presenting two projects actually today. So my friend Megan Faedron is currently studying abroad in Ecuador. So she was also working under Les Kaufman. Our projects are different, but they come from sort of the same premise and the same sort of focus. So specifically, my project was looking at the predator point of view to map out the trophic structure of the Gulf of Maine for policy. What Megan was doing was specifically looking at the different prey content, nutritional content of the prey species in the prey guild that I was examining for my project. So she used FAME analysis, which stands for fatty acid methylated ester analysis. Basically what she was doing was she was extracting all the lipids from all the different prey species of the Gulf of Maine and determining the concentration of mono and polyunsaturated fats in each of the species, because those are the most readily available forms of energy for predators in the system. So she was looking at the nutritional content of these prey and then using that data to hopefully help inform fishery policy. One specific issue that this data is going to provide data for, is that she found that there was a very high fatty acid content in a particular species of fish called sandlance, which has been showing drastic shifts in distribution in Stellwagenbank in the Gulf of Maine due to climate change, and that doesn't bode well for the whale watching industry. Just in 2012, whales were found way offshore, way down south, so the distribution of them is already affecting the industry. They're also thinking of opening a sandlance fishery on Cape Cod to sort of support the failing anchovy industry in Northern California. So this data is really providing information about what prey species are most important for the overall functionality and which ones are most vital to consider when considering new fishery policies and implementing them. And her poster is right next to mine, so I will be hovering kind of in between them if you guys have any questions. So it was a cool project. Feel free to come check it out. Great.
Hi everybody, my name is Vito Nath. I am a member of the computer science department, and under the guidance of Andre Lepetz, I have created my own programming language, which I'll be planning on expanding later as time comes along. Programming languages are very similar to spoken languages in that they have a certain syntax, they have certain rules, and majority of the population acknowledges them, but only a very few specialize in them. And so my whole goal with my programming language is not to create another language which powers databases or the newest websites, but really educates people in understanding what are the primary concepts in computer science and in programming, and how the possibilities and potential that can be found through programming are really actually accessible to everyone rather than the very few people who sort of really deal with them. And so what I had to do for my project was create three main components and one sort of user component. The three main components are called a tokenizer, a parser, and an interpreter. And we do this kind of in our head when we see written language and spoken language, but at a very, very rapid level. When we say something, those words are broken down in our mind and the meaning is given to each of those words. Combined, they give the full meaning of the sentence. So when I say my name is Vito Nath, each of those words has a specific purpose in that sentence. And very similarly in programming languages, every command that's typed in the computer has its own purpose. And so the tokenizer is the part which breaks it apart and gives each word its own sort of its own separation. The parser takes of those words and compares it to the language syntax, which I had to develop myself. And the interpreter is the one that says, okay, now I know, getting from the parser, this is what it says in the syntax. Let me flip the bits in the computer, in the hardware, and make the code execute. So for my presentation, or for my poster, I have my laptop out, and if any of you guys know how to type out the word print and write out any number, you too can become programmers. So please come check it out. Thank you.
My presentation was done under the help of Professor Brooke Blower, who's in the history department, and I really was interested in studying the importance of like humanitarian aid, international humanitarian aid during the Spanish Civil War, because it's a subject that's rarely studied. So a little bit of background on the Spanish Civil War. In 1936, it broke out in Spain between nationalists who were under Franco and loyalists who were kind of defending the broken democratic government. And so this war was extremely politically charged, and for that reason, not a lot of governments were able to provide neutral humanitarian aid. And there was one group that was really able to provide this neutral aid that was very essential in basically helping a lot of children and mothers get the food that they need while this war is kind of going around. And that was the Quakers, who created this group called the American Friends Service Committee. Now, the American Friends Service Committee was created in World War I, and really this group was basically created so that Quakers could avoid the draft, because they were conscientious objectors. They didn't want to be in the draft. But in the Spanish Civil War, I argue that the group really changed, and it was not so much focused on objecting to war. It was more focused on providing relief during the war. And so they actually were actors and participators in bringing that essential relief, even though they're a lot of times perceived as conscientious objectors to war. And so I also look how that drastic change during the Spanish Civil War translates to them during World War II, really bringing essential aid in France to people who are in concentration camps, to refugees there. And so they were able to provide a really large-scale program organization there. So I also learned a lot of really cool stories about some really heroic people. So yeah, come check it out.
So my project, first I'm really just going to talk to you about the motivation. So what are rare earth elements and why should I care? If you remember on the periodic table, all the way down at the bottom, separated, there were a group of elements that you could see, not the radioactive ones, but the other ones. It's a group of about 15 elements that are really important in terms of most high technology and renewable energies because of their use in terms of catalysts, phosphors, and permanent magnets. So if you were to say, well, why do we need to recycle them? Why not just cut down on our use of them? They're really in all of the big growing industries. So anything that has a screen on it, or anything that'll help you extract energy without having to use a fossil fuel. So because of that, their annual demand is probably going to be around 7.6% over the next 25 years. And as a non-renewable resource, what can we do in order to help maintain these stocks? Because right now there's really no effective recycling method for these, just because of the low concentrations that they're used at. It's not like you can just, open up my iPhone and take out a block of rare earth elements. It's just spread out throughout the entire thing. So I work for a professor of chemistry, Karen Allen, and what we tried to do is design a protein that will help us take these out of solution. And so, long story short, it hasn't worked yet, but if you check out my poster, you'll see really what was my first experience with grappling with protein structure and protein engineering.